Thank you very much. So first of all, thank you so much for organizing this wonderful meeting. So a big shout out to all the busy people that were, organizing all of this. And since my fellowship was a bit ago, I'm not really going to talk about what we did during this fellowship, but what we've been doing in the last few years. So, and I thought, what better to talk about after having, I saw quite a lot of cakes and cookies out there. So let's talk about obesity and diabetes for a little bit. So, having said this, I think we all know that obesity is a problem, and associated with the rising rates in obesity and especially childhood obesity, there's an increase in global rates of type 2 diabetes. And type 2 diabetes is a very complex multifactorial disease. And I'm not really going to talk about the genetics and everything, but it's important to keep in mind that it's really obesity, the lack of exercise, and of course multiple other environmental factors that contribute to the development of type 2 diabetes, in addition to all the genetic predispositions that we have. So one primary goal is always that weight reduction is an essential pillar of diabetes prevention and treatment. So having understood this, then of course the next question is, how is actually body weight regulated? And what I learned is never argue with physics. So when we look at the body weight regulation, it actually follows very basic thermoregulation, laws of thermoregulation, where if we translate it to metabolism, it says essentially that the energy intake equals the amount of energy that we expend, and the rest is going to be stored. Yeah? So that's as easy. So if we eat more than we expend, we're going to store the rest, and we store it mainly in adipose tissue or fat. So over the years, mainly driven by GWAS studies and other things, we understood which organs are preferentially involved in regulating these different elements, and it's really the intestine, of course, and the central nervous system that regulate the food intake side of things, whereas the skeletal muscle is predominantly involved in energy expenditure. So in mammals, or in small rodents especially, there's also brown adipose tissue contributing, but the role in humans is a little bit more questionable, I would say. So that, of course, leaves our most favorite organ, the adipose tissue, really as a passive bystander. And even though everyone associates adipose tissue with obesity, it really has very little to do with the regulation of body weight. However, what we know is that there is a point when the storage capacity of adipose tissue is exceeded and we get dysfunction in the adipose tissue, and this is then really inducing a cascade of events resulting in what is summarized as the metabolic syndrome, so certain types of cancer, diabetes, and cardiovascular disease. So it's not the body weight regulation per se that is impacted by adipose tissue, but it is the development of the metabolic diseases associated with obesity. So the easy answer to then the obesity problem is, if the energy balance is right, so just eat less and exercise more. Huh? And there's a truth in it, because this is a summary of a lot of different clinical studies where, depending on how little people eat and how much they exercise, they're going to lose a lot of weight, and this is what you can see in these dark blue bars, essentially. However, the truth is also that when you let these people go on and live their life, so don't supervise them anymore, pretty much all of them after 5 years regain the weight that they had lost, which is what you can see in these light blue bars. So that is the reason why there was, of course, a lot of interest from the pharmaceutical industry to develop weight loss drugs. And I think pretty much everyone by now has heard of all the breakthrough results that were achieved with this new class of hypothalamic targeting GLP-1 agonists. And this is just an example of a GLP-1 GIP agonist, which is called tirzepatide. And what you can see in this graph, these are clinical data, that when people take these drugs, they lose quite a significant amount of weight, which is correct. However, there are three more things that one needs to consider. First of all, this weight loss is not only adipose or fat mass loss, people also lose lean mass, which is a problem. The second thing is, as you can realize looking at this black curve, there is a plateau phase. So you don't keep on losing weight until you're completely lean for reasons that we don't understand. At some point, the drugs don't really continue to work. And this is then causing the other problem is because this frustrates people. and it's expensive, so they eventually going to get off this drug, so discontinue them. And then what happens, and this is what you can see in the light blue bar, is there is a weight regain. As if you stop exercising or dieting, then people regain weight. And here is really a big problem associated with this, because these are recent data from collaborators from us, and there are many other studies where we can show that there is an epigenetic memory of this past obesity that remains in adipose tissue, meaning that the adipose tissue remembers that you had obesity in the past, and this is primarily because there's an increase in inflammation and tissue fibrosis. Yeah? So tissue fibrosis is really an excessive accumulation of extracellular matrix. And we try to contribute a little bit to the understanding of the role of tissue fibrosis, not by studying the extracellular matrix, because there are so many components and so much redundancy, and I'm a mouse geneticist, so when we knock out one component, most of the times it's compensated by something else. But we decided to target the main cell adhesion receptors, which are integrins, to look what the role specifically in adipocytes of these cell matrix interactions are. However, as you can see in this graph, there are also quite a lot of integrins also expressed on adipocytes. So it did a trick using something that that I found in my PhD actually, which is a little here it's just a green ball protein. It's an intracellular adapter protein, which is important for the inside-out activation of integrins. So when we knock this protein, it's called kindlin-2 out, integrins are essentially inactive. And this affects all classes of integrins that are expressed in adipocytes. And by doing so, we could study the effect on adipose tissue. But what I wanted to show here is what we could find is that actually integrins form a complex with the insulin receptor. And the insulin receptor is really important for adipocyte function. And what we could further show is when we inactivate integrins, that this results in a reduction of the downstream signaling of the insulin receptor, as you can see in this graph. So in blue is the wild type, and red is the knockout, and on the right side you see what happens when we stimulate these cells with insulin. And while we get a nice response in the wild type cells, it was really blunted in the knockout cells. Essentially showing that one element how fibrosis acts on the adipocytes is that it really also already blunts adipose tissue simply because you change the extracellular matrix, and this also has an impact on the activation state of the adipocytes. The other important thing to mention here, however, is that we still need this hypertrophy of the adipocytes, so the growing in the size of the adipocytes, to cause trouble. Okay? So we wanted to study a little bit more what's going on, what are the regulators, and what can we learn about the fatty acid uptake in the adipocytes, which is really the main driver of size expansion in an adipocyte. And the way how we tackled this, like we did in all the years before, is we have a primary focus on cell surface proteins, because we think that they're really a central hub of organizing extracellular cues and organizing how the cell responds to this. Yeah? And we had published this actually still as part of the fellowship, this paper where we found some surface markers of adipocytes. However, one element that we did not publish at this time was a small two transmembrane protein, which is called NREC, nutritionally regulated adipose and cardiac protein. Because we found it to be really specifically expressed in adipocytes. However, the problem with this protein is it had no homology to anything. So there was no idea what this thing could do. The closest homolog is a bacterial channel, which didn't really help us much. However, we then got mice, we generated an antibody, we then did some proteomics to look for interaction partners, made a lot of mutations and did some molecular modeling. And what we could see is that this protein, the NREC is in green, you see a large cytosolic domain, then two transmembrane domains, actually interacts through a transmembrane interaction with CD36. And this is interesting because CD36 is the main long-chain fatty acid transporter, not only in adipocytes, but in most cells in the body. However, remember, only adipocytes express NREC. So we then looked at the knockout phenotype, and what we could see is in wild type is black or gray, and the knockouts are shown in red. is that when we do a lipid tolerance test, so gavage mice a lipid bolus and look how long does it take until it's cleared out of the blood, we could see that knockouts had a faster clearance of lipids from the blood. The triglycerides were lower in the serum. And when we measured actually fatty acid uptake into adipocytes, we could see that these cells take up more fatty acids. And this resulted in the end into an enlarged size. I hope you can see this. It's an H&E staining of two different depots. On the left side the wild type, on the right side the knockout, and the cells are just larger. Yeah? So that was, and this was all done on chow diet, on regular mice fed and normal diet. Yeah? So we then wanted to see, okay, how does it work? So we did cell culture experiments and just stainings, and we co-stained with NREC, caveolin 1 and CD36. Oh, this is what I forgot to do, because CD36 is not really a transporter, it's more a translocase, because it binds fatty acid in this endocyte host, and it's thought to be via caveolin-mediated endocytosis. And what you can see, hopefully, here is that in the wild-type cells, you have surface localization of NREC, caveolin, and CD36. However, in the knockouts, of course, there's no NREC, but while caveolin is still at the cell surface, most all of the CD36 was internalized. Yeah? And we could also show this dissociation by immunoprecipitation and other organs, but by the end of the day, what we could show is that when we inhibit using chlorpromazine clathrin-coated endocytosis, we could restore in the knockout CD36 cell surface localization, meaning that what happens is that while in normal cells there's a caveolin-mediated endocytosis, when we knock out NREC, there's a shift from caveolin-mediated endocytosis to clathrin-mediated endocytosis, which is known to have a higher turnover, which you can also see because there's more intracellular CD36. So that was the moment when my, at that time, student, now postdoc, was really excited, because on chow diet, cells are bigger. When we feed these mice now a high-fat diet, they're going to get really sick, and he already could see all his super papers published on this. As science is, we feed these mice a high-fat diet, and there was absolutely nothing. I don't show you the data. No matter what we looked at, these mice were completely normal. We took a vacation. A couple of weeks, no. Actually, he did, but no. It turned out to be really informative for us, because, again, for time reasons, I'm not going to go through the data, but I'm just showing you a cartoon, because what turned out to be true is that when we have a high-fat diet or a high extracellular fatty acid concentration, this results in a proteolytic degradation of NREC, so essentially phenocopying the knockout. Yeah? And then this results in a shift from caveolin to clathrin-coated endocytosis, increasing the fatty acid uptake. So in summary, I think what we found is that this small protein, but it is really only expressed in adipocyte, acts as a rheostat regulating fatty acid uptake in adipocytes, meaning that when fatty acid levels are low, it allows the adipocyte, allows other tissues like the heart that need to run on fatty acids to take this up. Whereas if fatty acid levels are really high in the circulation, then I will talk faster. If they are really high in the circulation, then the floodgates open up, and essentially the adipocyte clears out as much fatty acid as possible to prevent lipotoxicity. Yeah? So, adding to this little cartoon, that maybe also we need to consider the dietary patterns or what we eat also matter. Yeah? That leaves us with the last point that I'm quickly going to go over is the lipolytic side of things, you know, because not only lipogenesis or lipid uptake is important, but also if the adipocyte breaks down lipids. And one thing that many people don't know about insulin, it's very well known to induce glucose uptake, but when you look at this graph, actually the suppression of lipolysis is much more potent, or insulin is much more potent, about 6-fold more potent in suppressing lipolysis than in inducing glucose uptake. Meaning, way before glucose is cleared from our blood, we suppress lipid breakdown in the adipose tissue. And the reason why I mentioned this is because we stumbled over, I need to go really quick over this, when we wanted to study an enzyme in beta cells. Doesn't really matter for now what it is, what it does, but what we saw is that these mice have a basal hypersecretion of insulin. So there's no glucose, no difference in glucose-stimulated insulin secretion, but there's just more leakage of insulin out of these beta cells. And this was enough to shut down lipolysis and induce an hypertrophy. However, in this case, that was actually good for the metabolism, because you can see when we do a glucose tolerance test, the knockouts are again in red, then the blood, the glucose is cleared faster, than in the wild-type mice, simply because I think all the lipids are maintained in the adipose tissue where they should be. However, when we feed this mice a high-fat diet, we get an increase in hypertrophy, so cells get even bigger, and this is then inducing really insulin resistance. Yeah? So, indicating that elevated basal insulin secretion suppresses lipolysis, resulting in adipocyte hypertrophy and the predisposition to the metabolic syndrome. And we think this is interesting because when you remember the graph that I showed before, the levels of basal insulin secretion that humans have is actually overlapping with the amount of insulin that we need to suppress lipolysis. So, it could be that this is just describing some of the different responses of individuals to diets. And the last thing that I wanted to say is, because we want to say something also about the frontiers and what we want to do next, and how one of the issues that we are trying to deal with and which we are having problems bringing our basic science findings with conditional knockouts into the clinic or something, is that often we have these wonderful drugs or pathways, but we cannot use them because they are great in the tissue we want to target, but they have very unwanted side effects in other tissues. So in the shadows, in the last 10 years, we've been working on trying to figure out how we can create tissue-selective drug delivery vehicles. I just want to know, because after really a long time, I think now we cracked it, and we have some really cool aptamers, so single-stranded DNA, oligo platforms set up in the lab where we can now already target various organs and really conjugate those things to deliver targets or small molecules as RNAs to these tissues. And with this, I would like to thank my lab's members, all our collaborators, and of course you for your attention. Thank you very much.
Thank you very much.